Hi, this is Advances in Hemodialysis Part 2. Before we going to the second part of the advances in hemodialysis, we should think about if we believe in the history, we should realize the future. What about dialysis 70 years ago? It was based on very preliminary machines, very limited technology. Right now, it's expanded to more technology, more safer dialysis could be achieved. So in the future, what about the future? The bioartificial kidney and the implantable devices. The future towards a more continuous and physiological therapy to maintain renal endocrine function, to have both filtration and secretion. This could be achieved by implantable devices, which will be more continuous, more physiological, have some other renal function, and to have a filtration and secretion, we should have a renal tubule assisted devices. Bioartificial kidney, the theory and the promise. Bioartificial kidney means that placing renal tubule cells, primary cells, or cells lines established from various species on the inner surface of a hollow fibers. Bioartificial kidney will have more function than regular hemodialysis. Secretion function, reabsorption, endocrine, as well as some metabolic function. Removing more toxins by the renal tubule assisted devices. The regular hemodialysis with hemodialysis membrane is blind, can remove only molecule according to the sieving coefficient of the dialysis membrane, depending only on filtration. While renal tubule assisted device has filtration and secretion. That's achieved by a cell lining seated on the dialysis membrane by renal tubules. Such renal tubules can uptake of protein-bound uremic toxins and removed. We put a brain inside the membrane. So the bioartificial kidney is a synthetic porous hemofilter membrane seeded with renal tubule cells. Such a filter hemofilters membranes and seeded by renal tubules. So the bioartificial kidney can uptake protein-bound uremic toxins by the receptors of the renal tubules. 3 layers could be identified here. First layer is a hemofilter membrane. Second layer is a monolayer with tight junction from the renal tubules and a surface coating that can have the ability to attach the renal tubules to the hemofilter membrane. So, simply said that the future with bioartificial kidney is just putting a brain to the membrane. The bioartificial kidney, the hemofilter membrane seeded with renal tubules, can have many functions. A monolayer cells, has its own receptors, organic anion transporters, that can uptake and secrete diuremic toxins. If you look for the renal tubule devices with organic anion transporter, can uptake, for example, molecules through the receptors, moving such molecules toward outside or the filtrate. This type of therapy could not be achieved before, as such molecules are very big to be filtrated. So we have an uptake, secretion toward the filtrate. For example, if a filtrate is passing through the renal tubule assisted device through the blood flow, membrane layer, and filtrate through a renal tubule device can handle. However, the problem is sometimes the outer layer clogging by proteins and cells that needs more attention for the future technology. Patient has here two types of therapy: a hemofiltration therapy followed by a bioartificial kidney. So the filtrate can pass through the bioartificial kidney to eliminate more toxins, layer of blood, and the ultrafiltrate pass through the proximal renal tubule cells. How we can get the renal tubule cells? The source and the supply of such cells is not an easy. Either isolation, in vitro expansion of specific cells from the adult tissue. Cells of choice must have stem cell-like character, has also high capacity for self-renewal, ability to differentiate, develop the correct functional component. In the past, it was used by adult big kidneys, renal proximal tubules, progenitor cells, cells to be seeded into the renal assisted device. However, the ability of porcine endogenous retrovirus to infect human cells is important. So big cells are not used right now. On the other hand, human renal epithelial cells, isolated from kidney transplant discards, cells to be seeded into the rat system and will perform well with respect to viability, durability, and physiological performance. So pack requirements: homogeneous monolayer, porous membrane like polysulfone or polyether sulfone, coating to the membrane for cell attachment, such like pronectin, laminin, collagen. You need renal epithelial cells, and could be demonstrated by different stains inside the hemofilter membrane. You can find that the microvilli expression on the cell surface and this cell monolayer in the hollow fiber itself. The synthetic membrane has two parts, the inner and the outer part of the membrane. It differs in the pore diameter. The inner side has a small pore diameter, while the outer side has a bigger pore diameter, either like regular high flux membrane or high cut-off membranes. So in the outer parts, the pores are bigger, while the inner part, the pores are smaller. Seeded renal tubules on the inner part of the membrane has a capacity to remove toxins by its own receptors and could be seeded inside the fiber lumen, well identified by staining of such nuclei inside the fibers. As well, it's identified that Madden-Darby canine kidney cells from a polarized epithelium on the inner surface of the membrane. This membrane could be polyether sulfone, polysulfone, or others. And a confluent monolayer of these cells on the inner surface of the membrane, targeting the bigger molecules. The immune staining for tight junction proteins in green. This tight junction of renal cells appear lately, nearly after a week of incubation of the coated flat synthetic membranes with the renal tubules. So in diagrammatic presentation, renal tubule assisted device in series with a hemofilter. So if we can compare the kidney and the bioartificial kidney, we have a hemofilter like a glomerulus for hemofiltration, a bioartificial kidney seeded by renal tubules like renal tubules, a blood flow first through the hemofilter, then pass through the bioartificial kidney. The renal tubule assisted device is an extracorporeal device with a standard hemofiltration cartridge covered by a non-autologous human renal tubular cells along the inner surface, flow through a hemofilter, then flow through renal assisted device. In this device can metabolic products and reabsorption capacity can take place. So the flow can be from the patient, a blood flow with a hemofiltration, blood flow first through a hemofilter, part of the returning blood pass through the renal assisted device and returning back to the patient. Initial clinical results of the bioartificial kidney containing human cells in ICU patients with acute kidney injury from around 12 years ago had showed that a promising therapy. Patients with multiple organ failure and acute kidney injury had dialysis session with a hemofiltration in series with renal tubule assisted device. The results were promising with decrease in the granulocyte colony stimulating factor, interleukin 6, interleukin 10, as well interleukin 6 over interleukin 10 ratio. However, in this initial study of 10 patients demonstrated safe use of renal tubule assisted device therapy for up to 24 hours. Under RAT therapy, an actual mortality rate of 40% was demonstrated. The RAT treatment resulted in significant decline in granulocyte colony stimulating factors, interleukin 6, 10, as well the ratio of 6 over 10 interleukins. RAT therapy improved the 28-day mortality rate from 61% in conventional hemofiltration to 34% in the RAT-treated group. However, the clinical trial was halted during the phase 2B interval analysis. That's because of the suboptimal clinical protocol design and several fabrication and manufacturing process of the renal tubules. Accordingly, still seeking for a suitable supply of the renal tubules, one of them is the BioArtificial renal epithelial cell system. The BioArtificial renal epithelial cell system using a hemofilter with a high cut-off value of 65 kilodalton, a flow through the BioArtificial renal epithelial system, as well through an immune filter. This immune filter likes an immunoisolation filter, which protects the patient from any large molecules, byproducts, and or released by the BioArtificial renal epithelial cell system that could cause an adverse reaction. These BioArtificial renal epithelial systems with human renal epithelial cells on a disc derived from the enhanced propagation method of a cell expansion and could be a very good supply of the renal tubules necessary for seeding into the hemofilters. The bioartificial kidney steps toward clinical application include FDA approval for the first in-human clinical trial testing, where renal cell therapy has demonstrated safety and efficacy in the preclinical stages and in clinical studies. bioengineered cell system for mass production is required. Subsequently, a compact bioartificial renal epithelial cell system has been developed for use in renal as well other inflammatory, chronic and acute indications. Also, the bioengineered cell system should be capable for on-site, on-demand, whenever used in clinical. So the bioartificial renal epithelial cell system can be cryopreserved for transport as well for storage and reconstitution at the clinical sites. So that's what we had been looking for, to have such therapy and requirement available for clinical use. Toward a bioartificial kidney, will embryonic stem cells be the answer? The human embryonic stem cells could differentiate into functional renal proximal tubular-like cells. And in a study, it shows that there is a response to parathyroid hormone, as well gamma-glutamyl transferase activity, which measured by the concentration of the p-nitroaniline produced by the cells. Also for the ammonia production against control, and water transport assay. So it is promising line of therapy to have the embryonic stem cells, which can differentiate into a functional renal proximal tubules. The cultured blastocytes and the isolated cell mass, then the cultured human embryonic stem cells could have a specialized cells at the end, which is availability of efficient differentiation protocol. This cell therapy is successfully engraftment and has some maturation level. Durability and feasibility is still under questions. However, this cell therapy could be a bioreactor and could be used for the bioartificial kidney and others. Although encouraging, there were some limitations in the model described. In general, such cultures of the terminally differentiated human proximal tubules had greater function preservation when compared with the cells derived from the human embryonic stem cells. The problems are some downregulation of several transporters, including apical and basolateral glucose transporters, and organic acid cation and peptide transporters. So it lost some of the function. Additionally, a proportion of these cells de-differentiated in vivo experiments, and hence further tumorigenesis studies and epigenetics characterization are important if clinical use is to be used in the future. So understanding the bioactivity of stem cells seeded on the extracellular matrix scaffolds produced from discarded human kidneys, this is a very critical step toward a new generation of bioartificial kidney. The implantable device. At the University of California, San Francisco, researchers unveiled a prototype model of the first implantable artificial kidney that's on September 2010. They received $3 million in funding as of October 2012. and plan to go through human clinical trials by next year. The implantable device. The implantable bioartificial kidney builds upon the existing extracorporeal renal assisted device, which is a bioartificial kidney that combines both a membrane hemofilter and a bioreactor, as we discussed before. This bioreactor of human renal tubule cells is to mimic many of the metabolic, endocrine, and immunological functions of a healthy kidney. The implantable device. The ultimate goal of the kidney project is to apply micro-electromechanical system and a nanotechnology that can have the extracorporeal renal assisted device, and both are a surgically implantable, self-monitoring and self-regulating bioartificial kidney. So, through a micro-electromechanical system with a nanotechnology filter and renal assisted device, surgically implantable, can have a continuous function inside the body. After single surgery to establish a permanent blood connection, the bioartificial kidney processes blood continuously for 24 hours per day, which mitigates the inconvenience and the morbidities associated with intermittent hemodialysis. 3 key technology developments were needed to implement these functions. Number 1, a high-efficiency ultrafiltration membranes. Number 2, to control of blood materials interaction as a foreign body, such as thrombosis and protein fouling. Third point is to stable differentiate function of renal cells in an engineered construction. There is advances in silicon nanotechnology were required to make it possible to mass produce reliable, high porosity membrane, robust and compact membrane. That's a requirement for filtration. Improvement in molecular coating that impart blood compatibility and techniques to coat silicon membrane without blocking pores were needed. So we have a silicon membrane which are highly porous and compact based on a nanotechnology and coated. Then cell sourcing and storage issues had been resolved. All of these technologies are now in place. 2 components here, the hemofilter and the cell bioreactor. Component 1 is a hemofilter. Component 2 is a cell bioreactor. Hemofilter compact membrane relies on the body blood pressure to perform filtration without needing of blood pump or a power supply, just arterial pressure. Then a hemofilter must be capable of generating meaningful ultrafiltration, so it is porous at a given pressure, which is the arterial pressure, similar to capillary perfusion pressure. So you maintain the blood flow without need of a pump through a nanotechnology silicon membrane to produce a meaningful ultrafiltration volume. Then the second step is the cell bioreactor. The cell bioreactor is an advances in field of tissue engineering to grow and to maintain renal tubule cells. The bioreactor must be capable of high volume salt and water reabsorption from the ultrafiltrate, while maintaining a barrier to reabsorption of toxins. So we have 2 steps, a hemofilter for filtration and a cell bioreactor for reabsorption and other function. This small device is implanted in the pelvis, consists of a super thin silicone nanofilters, and it removes toxins, salt, and water from the blood with inflow and an outflow pathway, implanted in the pelvis, and is surgically implanted in the body, so it can produce urine in the bladder as well. Such silicone nanoporous membranes are part 1, and part 2, compartment 2, is the renal assisted tubules. Removes toxins, salt, and water from the blood. Implantable device at the University of California, San Francisco, giving the smallest device that can be surgically implanted with 2 components and a blood inlet and a blood outlet inside the device. The promising implantable device with 2 compartments is still small, smaller than cup of coffee, with blood inlet side and a blood outlet side. There are 2 components here. Compartment 1 with a nanoscaled filters to remove toxins from the blood with a super efficient membrane. Blood inlet flow depends on pressure from the capillaries, so it uses arterial pressure, not a pump. Then filling the silicone filters to produce filtrate. Then on the second part, the biocartridge, blood flow, then return back to the blood. The second compartment holds live kidney cells that perform the other biological actions of a real kidney. Inflow through silicone filters and outflow through a biocartridge. Such nanoscale filters are very promising and produced by nanotechnology. So implantable device with a hemofilter and a cell bioreactors consist the components of the implantable device and the kidney cells here to work. The hemofilter, incoming blood with super efficient membrane made with silicone nanotechnology, ultrafiltrate separated from the blood, contains dissolved toxin as well water, sugars, and salts, while cell bioreactor contains live kidney cells, can reabsorb much of the water, sugars, salts to the bloodstream. The toxins and excess water are passed into the waste outlet connected to the urinary bladder. Compartment 1 and compartment 2. Fluorescent microscopy image of the human renal proximal tubule cells cultured from discarded renal transplant kidneys on a silicone nanopore membranes with blue stains referred to cell nuclei, green referred to tight junctions, and red refers to cilia. In conclusion, the current status of bioartificial kidney, although studies about cell therapy and the wearable device have not been conducted for quite a long-term period, however, it's promising that these new therapies will be available as options in the near future. It is not a daydream that new therapeutic options such as BioArtificial kidney and wearable dialysis device for end-stage renal disease patients will be applied in the routine clinical practice in nephrology area. Thank you very much.